gentlemen, welcome to another interesting live webinar. And this time the topic is molecular genetics and diagnostics tool. We assure you it's going to be another interesting webinar under the aegis of Mumbai Hematology Group. And we at INTAS are extremely proud of this collaboration. The gentleman on the screen is our program director, none other than Professor Dr. M.B. Agarwal, sir, President of Mumbai Hematology Group, Professor and Head Department of Hematology at prestigious Bombay Hospital and Lilavati Hospital and Bridge Candy Hospital. We all know him very, very well, teacher of teachers, an institution in himself, and his tireless effort has brought in so many research work and research speakers. So may I request you, sir, to take it forward from here, Professor Dr. M.B. Agarwal, sir.
Thank you, Dr. Manoj, for those kind words. Good morning to one and all if you're from India. Good afternoon, good evening if you're from the rest of the world. Today, our guest speaker is Dr. Sanjeev Kumar Gupta from New Delhi. He will be lecturing on molecular genetics of B acute lymphoblastic leukemia, recent updates and diagnostic tools, a subject which is very close to his heart. This webinar is brought to you by Mumbai Hematology Group, supported by INTAS and managed by Perfect Square. I thank Dr. Manoj Kumar and his team from INTAS, Mr. Yash, Mr. Kalpesh and the team from Perfect Square, Executive Committee of Mumbai Hematology Group, our guest speaker, Dr. Sanjeev Kumar Gupta from Delhi, all our discussants who are themselves eminent hematologists or medical oncologists, you participants for sparing your Sunday morning, afternoon or evening. Next weekend on Saturday at 7 p.m. onwards IST, we have Dr. Nicola Conran from Hematology Center, University of Campinas, Brazil. She will be speaking to us on emerging pharmacotherapeutics approaches for the management of sickle cell disease. And next Sunday morning, 11:30 a.m., we have Professor Fallini from Italy speaking to us on 2023 update on T-cell lymphomas. Our discussants today are put up here alphabetically. We have Dr. Akshaya Mandloi Goyal from TMH Varanasi, Dr. Ankur Ahuja from Army Hospital, Delhi, Dr. Deepak MB from MSMC Narayana Health City, Bangalore, Dr. Geeta Vidyadharan from Amrita Institute, Cochin, Dr. Jyoti Bajaj Sahne from GCRI, Ahmedabad, Dr. Mahesh Mandolkar from Dinanath Mangeshikar, Pune, Dr. Mohammad Taha Ali Khan from NIMS, Hyderabad, Dr. Mayak Marlar Shwe from University of Medicine, Mandalay, Myanmar, Dr. Nilesh Shah from NHL Municipal Medical College, Alice Bridge, Ahmedabad, Dr. Rupam Deka from Bhubaneswar Barua Cancer Institute, Guwahati, Dr. Savitri Singh from PGI and Child Health, Noida, Dr. T. Ramadevi from GCRI, Ahmedabad, Dr. Umang Patel from Green Cross Genetics Lab, Ahmedabad, and Dr. Vibha Bhavna from Bharati Vidyapith, Pune. And now coming to our guest speaker for the day, Dr. Sanjeev Kumar Gupta from New Delhi. He is Professor, Laboratory Oncology at Dr. B.R.A. Institute Rotary Cancer Hospital at AIIMS, New Delhi. He did his DM in hematopathology from AIIMS, Delhi, 2010. Earlier, he did MD in pathology from MAMC, Delhi, followed by DNB in pathology. He did DHR fellowship in the field of genomics from MD Anderson. His area of interest is molecular genetics of hematological malignancies, especially acute lymphoblastic leukemia. He was awarded with the AIIMS Excellence Research Award for 2017 for his work in this field. He was also awarded with AIIMS Oncology Research Award in 2019 for his published work in complementary role of molecular genetic profile in outcome prediction of BALL, in addition to the standard practice of flow cytometry-based MRD assessment. Recently, he was awarded with the AIIMS Oncology Research Award in 2021 for proposing the molecular genetic risk scoring MR+ in B acute lymphoblastic leukemia. He was a part of an international collaborative group studying the involvement and clinical impact of COBAL gene rearrangements in the B acute lymphoblastic leukemia cases with Icaros deletion. Earlier, he has worked on rapid diagnosis of acute promyelocytic leukemia using immunofluorescence. For this research work, he was awarded Mrs. Geeta Mittal Medal 2009 for clinical research at AIIMS New Delhi, followed by Dr. J.C. Patel Award for the best paper at Golden Jubilee Conference of ISHTM in 2009. He has worked on establishing the data on prevalence of other genetic variations in Indian B acute lymphoblastic leukemia, which are prognostically relevant, like intrachromosomal amplification of chromosome 21 and ETS-related gene deletions. He has evaluated the utility of various molecular diagnostic techniques like multiplex ligation-dependent probe amplification, RT-PCR, multiplex fluorescent PCR, Sanger sequencing, ARCA-based NGS, flow cytometry, FISH, etc., in detecting the genetic variations of B acute lymphoblastic leukemia. He's contributed towards maintaining the high standards of published research work through the peer review process and was awarded the Best Reviewer Award, Indian Journal of Hematology and Blood Transfusion at the 59th Annual Conference of ISHBT 2018. Currently, he is Associate Editor of the same journal. I invite him to give his lecture on molecular genetics of BALL, recent updates and diagnostic tools. Over to you, Dr. Sanjay.
Thank you so much, sir, for this kind introduction. And I thank Professor Agarwal and the entire organizing team of Mumbai Hematology Group for inviting me for this talk today, which is on molecular genetics of BALL, recent updates and diagnostic tools. I hope the screen is visible.
Yes.
Okay, so I'll start by saying that in all acute leukemia workup, we look at the clinical history and the background. And generally, we have a very short history of few days or few weeks. And the history contains generally of fever, fatigue, bony pains, or bleeding issues, or easy fatigability, easy bruisability. And when we look at the morphology, we find more than 20% blasts with high NC ratio in peripheral smear or bone marrow examination. And we do the cytochemistry also, for especially for myeloperoxidase, to see if these blasts are MPO negative or positive. Then we look at the immunophenotype. So all the residents, what they do, they send one sample for morphology and cytochemistry, one sample for immunophenotyping to look at the lineage of the blasts, and one sample is sent for the genetic analysis. Now the genetic analysis samples can be, one can be heparinized sample, which can be useful for the conventional karyotype and FISH-based studies. So the preferable sample is green top vial heparin. And the other sample is sent for the molecular genetics, the DNA and RNA analysis, for which EDTA sample is preferred. So the genetic studies like PCR, reverse transcriptase PCR, real-time PCR, Sanger sequencing or next generation sequencing can be done depending on the need and the resources available with different labs. So we will be mainly focusing on the genetics part of BALL in today's talk. So in next 45 minutes, I'll try to give you an overview about the common genetic changes in BALL, something about Philadelphia-like ALL, what are the new molecular genetic categories which have been recently described and included in the new WHO and ICC classifications of BALL. Then what are the copy number alterations and what is their significance? how these copy number alterations in BLL can be detected and then can be used for molecular genetic risk classification. And I'll be sharing our experience also in this area. Coming to the genetic pathogenesis, we know that in any leukemia, especially acute leukemia, there will be some initiating lesion or the driver lesion. For example, one of the recurrent translocations in BALL, like ETV RUNX1 or BCR-ABL, they can be the initiating lesion. After that, there is a developmental arrest of these altered cells by alterations in the B cell transcription factor genes, like paired box gene, Icaros, early B cell factors. And now these altered and arrested cells will proliferate with the help of other cooperating events. So there will be additional mutations in the cell cycle genes, tumor suppressor genes like CDKN or TP53, cytokine receptor genes like CRLF2, RAS signaling pathway genes, lymphoid signaling genes, transcription factor genes, and mutations in the epigenetic regulator like EP300. So by accumulating these further mutations, now these altered cells proliferate and lead to acute lymphoblastic leukemia. And further on chemotherapy, these mutated clones may accumulate, leading to relapse of ALL. So genetics plays an important role in the initiation as well as in the relapse of BALL. Now coming to the routinely tested genetic abnormalities in BALL. Now, BALL is genetically heterogeneous because we find both the numerical as well as structural chromosomal abnormalities in BALL. So the commonly tested structural abnormalities are the recurrent chromosomal translocations like BCR-ABL, ETV RUNX1, MLL gene rearrangements or KMT2A, and translocation 119. So we generally send the sample for RT-PCR based or FISH-based analysis of these recurrent translocations. And then aneuploidies are checked either by conventional karyotype, where all the chromosomes are spread, or we can do the flow-based ploidy analysis. Out of these translocations and ploidy changes, some are classified as good risk. For example, hyperdiploidy, more than 50 chromosomes, and ETV RUNX1 translocation. And we see that the prevalence of these changes is more common in children as compared to adults. So the good risk genetic changes are more common in children, whereas the poor risk genetics like BCR-ABL is classified as poor risk is more prevalent in adults, and the prevalence increases with age. And some are labeled as intermediate risk. So we can say that overall adults have a poor genetic profile as compared to children. But thankfully, BLL is predominantly a disease of children. So these are the techniques which are commonly used, like the multiplex RT-PCR. For that, RNA is taken because we want to know the fusion genes. We make the cDNA, complementary DNA, and then do the PCR. So in this lane, there is a ladder, and then we can see the internal control band and a band in this patient suggestive of BCR-ABL P190. In another patient, there is an internal control band and a band suggestive of translocation 1221. The same thing can be checked on FISH also. This is showing 2 red and 2 green normal pattern of ETV RUNX1 probes. This is the schema where the green color is coming from the ETV6, whereas the orange-red color is coming from the RUNX1 probe. So 2 copies of green, 2 copies of red is normal. If there is any translocation, there will be a fusion signal containing the yellow color with co-localization of green and red. So this is showing that co-localization or fusion signal, but this is the example of break apart probe. Now MLL rearrangements mean there are multiple partners of MLL. So either we look for each partner individually, or the better strategy is to use a break apart probe on FISH. So in FISH, these are the 2 major strategies: dual color, dual fusion, or break apart probe. So we see that the probe is coming from 5' end of KMT2A, the red orange probe, and the green probe is coming from the 3' end. So this means this fusion means the MLL or KMT2A is intact. If there is any rearrangements of KMT2A, there will be a splitting of this yellow signal into red and green. So we have to know what type of probe has been used to analyze the FISH results. Now in this part of the picture, we see that how flow cytometry can be used for ploidy analysis. So this is the gating of blast, which are CD45 DIM2 negative, which are CD19 positive. And then you look at the DNA content of the lymphocytes, blast, and then overlay to find out a DNA index. So this can be done using a dye, which is a FX cycle violet dye on the flow cytometry. And we get to know whether the DNA index is suggestive of hyperdiploidy or hypodiploidy or normal. In the same reaction, we can also add the antibody for CRLF2 to check for overexpression of CRLF2 in these 19 positive blasts. Conventionally, we see the ploidy changes being detected by the conventional cytogenetics. So that can also be done. But majority of cases of BLL, we will not find any of these recurrent chromosomal translocations or ploidy changes. So they will be negative for these changes and they will be labeled as B other ALL. So we'll find a majority of our cases fall in this group. And in this subgroup, because of the high throughput genomic techniques like next generation sequencing being available now, a lot of new genetic abnormalities have been described. So these are the new genetic subtypes, and they have been more common in especially adolescent and young adult cases of BALL. So we'll be looking at various terms like whole genome sequencing. That means that the entire genome is interrogated, including the coding area as well as non-coding area, the introns and exons. If we interrogate only the coding area, the exons, then the term used is whole exome sequencing. And for in BALL, the more useful analysis of RNA, the transcripts, where we can check the fusions. So the predominant cases are of fusion genes. So where we need the entire RNA being interrogated, or that is labeled as whole transcriptome sequencing. Then there can be targeted sequencing of all the interesting genes we can include in one panel and look for only those targets rather than entire genome or transcriptome. So now one of the first new subtypes was described almost a decade back, that was PH-like ALL, which was based on the gene expression profiling after all these RNA sequencing and gene expression studies by Children Oncology Group and Dutch Oncology Group. Simultaneously, they described this entity. So this is an entity which is having the gene expression profile very similar to PH positive cases, but actually there is no BCR-ABL translocation or fusion protein in these cases. So this is labeled as PH-like ALL. The prevalence increases with age, just like PH positive ALL. The highest prevalence being in 21 to 39-year age group at 27%. These cases also have a very high frequency of Icaros alterations. So Icaros alterations are not diagnostic of PH-like ALL, but they can be taken as a surrogate marker, and these cases can be looked for PH-like lesions. Overall, these cases are aggressive, having an unfavorable outcome. Now, PH-like ALL is not a single disease entity. It is again a group of disorder. It is genetically heterogeneous. And what we find that these cases may have ABL class fusions with multiple partners, erythropoietin receptors or JAK2 rearrangements, CRLF2 rearrangements, other JAK-STAT lesions, RAS pathway lesions. So this is a group of disorder mainly with fusions of these kinases with different partners. So as we see that these kinases are multiple fusion partners, and many of these can be targeted with the available tyrosine kinase inhibitors or JAK2 inhibitors or other inhibitors. So a lot of clinical trials are also undergoing to find the targetability of these PH-like lesions. The problem with PH-like lesion is that there is no consensus criteria for diagnosis. There is no consensus algorithm or definition. And each lab has to devise their own strategy. For example, this is the algorithm used by the Children Oncology Group. They use a low density microarray to screen all their BLL cases, and based on the analysis microarray score to select out the PH-like cases, then they look for CRLF2 overexpression in these cases. If CRLF2 is getting overexpressed, then they analyze these cases for CRLF2 and JAK mutation analysis by using FISH and PCR. If CRLF2 is normal, then they use the multiplex PCR of known PH-like fusions of ABL class and JAK2. And if these PCRs are also negative, then they analyze their cases with RNA sequencing, either by Archer panel or the Foundation 1 panel. The problem is that this low density array to screen initially is not available outside of the COG. This is not commercially available. So then what to do in this case scenario? Then one can look for the conventional cytogenetic changes, the translocations and the ploidy changes. In negative cases, one can go for flow cytometry overexpression of CRLF2. If CRLF2 is getting overexpressed, one can look for the CRLF2 rearrangement by FISH probes for PH-like ALL cases. If that is negative, we have another set of FISH probes for other ABL classes like ABL1, ABL2, PDGFRA, PDGFRB. If that is also negative, then one has to go for whole transcriptome sequencing. So it is a sequential approach which can be used to detect all the PH-like lesions. So the techniques which can be useful to detect the pH-like cases includes flow cytometry for CRLF2 overexpression. For example, here we see this red cluster, which is CD19 positive, CD10 positive, showing overexpression of CRLF2 or TSLP receptors as compared to the internal negative lymphocytes. We can use FISH break apart probes for ABL1, ABL2. Why break apart probes? Because these have multiple fusion partners. So any rearrangements of ABL can be detected by using a single probe. And then specific partners can be checked by further reactions. MLPA is useful to detect the cases with Icaros deletion, pseudo-autosomal alteration, CRLF2 amplifications. As we can see here, we'll see some examples. Multiplex RT-PCRs can be done for known pH-like fusions. So there are 9 multiplexes which can be done to detect the 50 to 60 known pH-like fusions. Then we can do Archer-based RNA sequencing. This is an 81 gene panel which is commercially available. And then we get to know the known and unknown fusion of the selected genes on this panel. If we want to know all the known and novel fusions of the entire genome, then we have to do whole transcriptome sequencing or whole genome sequencing in a case. This is a case where there is a deletion of EBF, early B cell factor, Icaros deletion, paired box deletion, along with amplification of CRLF2. And this is correlating with the CRLF2 overexpression on flow cytometry also. In these CD34 positive blasts, the red cluster is showing CD10 expression, CD34 and CRLF2 is overexpressed. Now, this is another interesting case where we did the FISH for BCR ABL break apart probe. This was a case of relapsed BALL and it belonged to the B other category with no specific cytogenetic change. So as we discussed in the break apart probes, the yellow colocalization signals are normal. So this is a normal cell. As we can see in the scheme, the probes are coming from the 5' end, the green probe, and the red orange probe is coming from the 3' end of ABL1. So these cells with arrow pointing towards them show splitting of one of the yellow signal into one red and one green. This indicates there is a rearrangement of ABL1 in this case. So then we wanted to know the specific partner of ABL1. And these are the 12 known fusion partner of ABL1 in the literature. So we did the multiplex RT-PCR, but it was negative with only the internal control bands being seen. Then we analyzed this case by Archer 81 gene panel, which is a targeted RNA NGS. And the advantage is that any partner gene of these genes on the panel can be tested by this panel. So for example, we are interested in ABL1 in this case. So on running this Archer NGS panel, we found that there is a fusion of sortin-nexin gene with ABL1. But then we realized this fusion partner was already tested in our known fusion partners of ABL1. This included SNAX2 gene also, but the PCR was coming negative. The reason was that in our case, the breakpoint was exon 9 of SNAX2 with exon 4 of ABL1. Whereas in the cases which have been described in the literature, the breakpoint is in exon 3 of this gene. So that is why the primers in the panel were not working for our case. So we designed the primers as per our breakpoint, and we did the RT-PCR to confirm this fusion, and followed by the Sanger sequencing also confirmed the fusion of sortin-nexin gene with ABL1. So if we find any new finding, then we have to do an orthogonal testing by another technique like RT-PCR or Sanger to confirm these novel fusions. So till now, in the initial analysis, we have done 24 cases of our high-risk BLL, and we have found these fusions in our BLL cases, like this case with a novel breakpoint, then P2RY8 and erythropoietin receptor fusing with IGH. There was another case of a novel fusion Icaros with class 2 transactivator in a case of relapsed BLL, and PAX5 ETV fusion. Then all the positive controls that we included in Archer testing like ETV rungs1, BCR-ABL, even cases with Icaros deletion could be confirmed on the Archer testing. Even in cases with low blast count, because in cases with less than 40% blast, MLPA may become false negative. So Archer is a useful technique to detect these pH-like lesions or fusions, especially for the 81 genes. Now coming to the other new molecular genetic categories which have been recently described and how the classification has changed. So the Mulligan group have analyzed almost 2000 of BALL cases using the RNA sequencing and other technique. And they have plotted this gene expression profile of the 1000 most differentially expressed genes as these two-dimensional Tisney plots. And we see that the cases with different genetic abnormalities cluster in different clusters. For example, this yellow cluster of cases are the cases with ETV-RUNX1 fusion. And the gray dots close to this cluster mean that the gene expression profile in these cases is very similar, but actually the ETV-RUNX1 fusion gene is absent. So these cases are labeled based on the gene expression profile ETV-RUNX1 like. Similarly, this blue cluster here is KMT2A or MLL gene rearrangements, and the gray dots which are close have a gene expression profile very similar, but KMT2A is absent. So these cases are labeled as KMT2A like based on the gene expression profile. Similarly, we have pH positive cases here, and pH like gene expression profile is very close. So that is how now we have a lot of new genetic entities being defined. Now we have 23 genetic subtypes, and they have been divided into 3 classes. Class A, which is based on the gene rearrangements. Class B, which is defined by the karyotype, the conventional karyotype, and type C, which has the new categories defined by the integration of expression profile, karyotype, and or genetic mutations. So let us see what are these 23 genetic subtypes. So in class A, we have most of the conventional fusions like ETV-RUNX1, BCR-ABL, TCF3-PBX. The new categories are DUX4 rearrangements, zinc finger 384 rearrangements in BLL, myocyte enhancer factor 2D rearrangements, cases of BLL with BCL2, MYC or BCL6 rearrangements, BLL with NUT M1 rearrangements, HLF rearrangements fusing with TCF3, that is the translocation 1719. Then the second category, the B class, is mainly defined by the conventional cytogenetics. So we have the normal hypoploidy, hyperploidy, along with intrachromosomal amplification of chromosome 21 in this category. Then in class C, we find a lot of new classes based on the gene expression profile, like pH-like, ETV-RUNX1-like, KMT2A-like, ZNF384-like also. So based on the prediction analysis of microarray score. Then we have other alterations like PAX5 alterations is a separate category. And specifically PAX5 PATR point mutation is given a separate category because of their different outcomes. Icaros point mutation N159 is also a separate category, which is different from the Icaros deletions. So we'll look at some of the salient points about the new genetic categories. And what we see that this is the international consensus classification, which has incorporated many of these new categories like the MYC, DUX4, MEF2D, ZNF384 as either the full entities, and some of these are provisional entities in international consensus classification. Even the pH-like has been divided into ABL1 class and JAK-STAT activated and NOS subtypes of pH-like. Similarly, in the WHO classification, we see in 2016 classification, these were provisional entities, the BCR-ABL-like and IAM21. Now, these entities have been upgraded to the full entities, and the new categories are ETV-RUNX1-like, BLL with TCF3 HLF, that is a translocation 1719. And the last category can encompass any emerging subtype based on the genetics, like B lymphoblastic leukemia lymphoma with other defined genetic abnormalities. So all other new categories can be included in this subgroup or any other new emerging category also. Now let us look at the salient points about some of these new categories. Now 5 to 10% cases of BALL will show DUX4 rearrangements and more common in adolescent and young adults. The feature on MLPA which can be detected that almost all cases will show ERG deletions. So we can use an MLPA kit P327 where we can look for these ERG deletion. These cases have a very good outcome even in the presence of poor risk feature, like Icaros deletion are independently a poor risk feature, but if they are present along with ERG deletion, then the outcome is better. There may be fusion of DUX4 with IGH gene also. Now for detection, we have to do RNA-seq for most of the fusions in BALL. We have to do the whole transcriptome sequencing, that is the RNA sequencing. We can also do the IHC for DUX4 overexpression in these cases, or we can do the real-time PCR for quantitation of DUX4. Another hint can be taken from the flow cytometry. Like we can use CD371, and these cases with DUX4 rearrangement show variable overexpression of CD371, which can pick up these cases with good prognosis. Now, 5 to 10% cases will show zinc finger 384 rearrangements, mainly the fusions with different partners. And depending on the partner, the outcome is different in these cases. For example, the most common partner is EP300, which is an epigenetic regulator. And the outcome is then good. As we can see in this green survival curve, both the event-free survival and overall survival is better with EP300 fusion. Whereas if the partner is TCF3, then the outcome is bad, as can be seen in this red survival curve. So depending on the partner, the outcome can be variable. Again, in addition to the RNA sequencing, we also have the break apart probes for zinc finger 384 rearrangements. Now, another interesting point about this category is on flow cytometry, we can get a hint. When a BLL case has a CD10 dim expression or absent, along with presence of CD13 or 33, that can be a hint for cases with ZNF 384 rearrangements. So there is a problem of lineage ambiguity in these cases. In fact, in children, if we see the mixed phenotype acute leukemia, almost 50% cases will show ZNF 384 rearrangements. Then the cases with myocyte enhancer factor 2D rearrangements are 3 to 5% cases, mostly in adolescent. And these also have a poor outcome. These cases have overall a poor outcome and resistance to chemotherapy. But there is a high expression of HDAC9, which is a target for MEF2D. And that can be used therapeutically, that this may sensitize the cell to HDAC inhibitors, which are available. For detection, we can do the RNA sequencing for fusions, or the break apart probes for MEF2D can be used. In these cases also, CD10 may be dim or absent, along with presence of cytoplasmic mu chains. Now, some of these cases of MEF2D rearrangement may show CD5 or CD33 expression. CD38 is uniformly expressed in all cases. Then 2 to 5% cases may show MYC rearrangements in BLL with a poor outcome. There may be fusion of MYC with IGH, or there may be BCL2 and BCL6 rearrangement, means there can be double hit or triple hit kind of rearrangements. And that can be seen in Burkitt's and other high-grade lymphomas also, where there are double hits or triple hits. But these cases of lymphomas will have a different mutational profile and evidence of somatic hypermutation. Whereas our BALL with MYC rearrangements, we will label when there is unmutated immunoglobulin variable chains. And there will be feature of immunophenotypic immaturity, like TDT will be positive in most of these cases. So for diagnosis, we can use the FISH using the MYC probes, BCL2 and BCL6 probes. Either we can use a strategy of break apart probe on MYC, so there is a splitting indicating MYC rearrangements. Or for MYC IGH fusion specifically, we can use dual color, dual fusion probes, where the yellow signal fusion probes will show, indicate the fusion of MYC with IGH. Another new category in 2% cases is NUT-M1 rearrangements. It is mainly seen in infants and children rather than adults. And depending on the age, the fusion partners may be SN1, BRD9, or CUX1, or ZNF618. So these are the different partner genes. Overall, they have a good outcome. So we can do the whole transcriptome sequencing, or NUT-M1 break apart probes can be used for fusions. Overexpression of NUT-M1 can be also tested with IHC or RT-PCR based technique. Now, this is another category where CD10 may be dim or absent with the expression of myeloid markers like CD13, CD15, CD33. So till now, whenever CD10 used to come dim or negative, we used to think of only MLL gene rearrangements in BALL. But now with these new subtypes, we know that differential diagnosis in a case with CD10 negativity can be even ZNF384 or MEF2D rearrangements, or even NUT-M1 rearrangements. So our differential diagnosis have increased. This is a very rare subtype. Less than 1% cases may show HLF TCF3 fusions. They have a very poor outcome. Like these are the translocation 1719 cases. So we can do the PCR or a FISH probe. There is a triple probe which is available containing PBX1, TCF3 and HLF. So one can detect fusion of 119 PBX3 with TCF3 or 1719 HLF TCF3. Like in this case, there is a fusion of this aqua probe with the green probe, HLF fusing with TCF3. So this can be the RT-PCR based analysis also. And these cases may show hypercalcemia. And some reports indicate there are remissions with blinatumomab and HSCT because they have bright CD19 expression. So anti-CD19 immunotherapy may work. Around 2% cases in children will show intrachromosomal amplification of chromosome 21. Now, these patients have poor outcome with standard chemotherapy, but if chemotherapy can be intensive, if intensive chemotherapy is used, then the reports have indicated that the outcome improves significantly. Now, what happens pathogenetically here is that generally there is a terminal deletion of chromosome 21q, followed by breakage fusion bridge cycle and chromothripsis. Now, what is chromothripsis? There are thousands of clustered chromosomal rearrangements happen in a single event. So what we see in chromosome 21q, there is alternate loss and gain pattern, which can be seen on MLPA or NGS or RACGH. On FISH, we find that there are more than 5 RUNX1 signals. So when ETV RUNX1 probes are used, the green color is coming from ETV6 and the red color is coming from the RUNX1 probe. So if there are more than 5 RUNX1 signal, especially if there is a clustering like this, that indicates there is an intrachromosomal amplification of chromosome 21. Okay, some other rare cases which have been detected, less than 1% cases where there is a fusion of upstream binding transcription factor with ataxin 7 like 3 gene. And simultaneously, there is a deregulation of CDX2 with overexpression of CDX2 in these cases. So there are cases with this fusion along with overexpression of CDX2 with a poor outcome. Again, we can detect it on RNA sequencing or RT-PCR based techniques. This is another subtype where CD10 may be negative with presence of cytoplasmic IgM. PAX5 alterations, as we discussed, have been divided into 2 groups. So one group has all the rearrangements, sequence mutations, and intragenic amplifications with multiple partners. But P80R type of PAX5 mutation is given a separate category because it has intermediate prognosis. In adults, it is even better. So in children, we see PAX5 P80R is slightly better off as compared to other PAX5 alterations. And in adults, this curve is very good as compared to other PAX5 alterations. So this is the PAX5 P80R alterations which are clustered here. And all other subtypes are classified in the PAX5 altered group. And the other rare subtypes include the Icaros point mutation N159Y with intermediate prognosis. There is a mutation in the DNA binding domain which increases the addition of these cells, just like in Icaros deletion cases also. And in many cases, there is gain of chromosome 21. And the last new category which is there is BLL with mutated ZEP2. There may be a point mutation or there may be fusion with IGH. And these cases also have a marked poor outcome, poor prognosis in less than 1% cases. And frequently we can find NRAS mutation in these cases. So those were the new genetic subtypes, and now they have been included in the ICC classification mainly and a WHO classification also. Now coming to the copy number alteration and what is their significance in BALL and how we can detect copy number alterations. So we are looking for copy number alterations in the genes related to B cell development and differentiation, like paired box gene, early B cell factor, Icaros, cell maturation gene, hematopoiesis, and cell cycle progression like CDKN, retinoblastoma. So these were first shown by the Mulligan group in almost 40% of their BALL cases. And these were mainly the PAX5, EDF, and Icaros. Now there is a commercially available kit, the multiplex ligation dependent probe amplification kit, which encompasses all these genes of B cell development and differentiation. And they can be tested on the DNA and can be plotted to look for the normal pattern, or if there is a pattern with deletion of one or many of these genes. So what is the principle of this technique, multiplex ligation dependent probe amplification? So basically it is just like a multiplex PCR that we do, but the limitation in multiplex PCR is that only 5-6 targets can be clubbed together. But in this reaction, 50 to 60 different targets can be interrogated in the same reaction. How that is possible is that the primers which are attached to these probes are same. The forward and reverse primers are same in all the different probes. So the probes are designed in two parts, the right probe oligonucleotide and left probe oligonucleotide. There is a stuffer sequence, and then there are primers at the end. So there is a hybridization of these probes, mixture of probes with the target DNA. After hybridization, there is a ligation step followed by PCR. And then the fragment analysis is done. So the different fragments coming from different targets can be differentiated based on the stuffer sequence length. So we see that this grain area is different in different probes, whereas the primers are same. So the PCR is PCR amplification is very easy because the same set of primer is used, and fragment analysis will tell us the different targets. So there is a sample denaturation, probe hybridization, ligation of the left and right probe oligonucleotides, followed by PCR amplification, capillary electrophoresis. So this is the electrophoregram, and then this is plotted as the probe ratios after the intrasample and intersample normalization. So this is indicating the normal pattern, and here we have the deletion of Icaros 4 to 7. So there is one probe for each exon, 8 exons of Icaros. So this is indicating deletion. The same can be confirmed with other kits with more probes per exon. Like this is a kit with 2 probes per exon in confirming the Icaros 4 to 7 deletion along with CDKN deletion. The additional investigation which can be done is breakpoint specific fluorescent multiplex PCR. So we can use the labeled primers for Icaros and we can check for deletion. This is more sensitive. And the other advantage of doing the PCR for Icaros deletion is that we can sometimes detect multiple clones, which can be missed on MLPA. So here the green peak is indicating the exon 4 to 7 deletion clone, and the blue peak is coming from the clone containing exon 2 to 7 deletion of Icaros. And these cases of Icaros deletions can also be confirmed with Sanger sequencing. So here we have fusion of exon 4 with exon 7 with some nucleotides intervening. So out of all these genes, particularly deletion of Icaros was found to be independently associated with poor outcome in BLL, again shown by the Children Oncology Group in this landmark paper. So when we analyzed our cases of Icaros deletion in this initial cohort of 101 cases, which included both adult and pediatric cases, the Icaros deletions were detected in almost 29% cases, which was much higher than the published literature. And we found that in BCR-ABL positive, it was almost double as compared to BCR-ABL negative. Similarly, adult cases had almost double prevalence as compared to the pediatric cases. And these cases were associated with induction failure rates to be higher. This is a more recent analysis of 320 pediatric cases, which are pH negative only. And there the 14.7% cases have BCR-ABL, negative cases have Icaros deletion. And these deletions can be of 2 types. Either there can be deletion of all the 8 exons of Icaros, leading to haploinsufficiency or loss of function, or there can be exon 4 to 7 deletion, which is leading to an IK6 dominant negative protein. But both these subtypes had poor event-free survival and overall survival in our cohort, and has been shown in multiple studies that Icaros deletion are associated with poor outcomes. Now, looking at the copy number alteration profile of all the 8 genes which are involved in BLL, development and differentiation, we found the 162 patients, which are both adults and pediatrics, 70.4% cases had at least one copy number alteration. And the common genes which were affected were CDKN, paired box gene, and Icaros gene. Especially the Icaros and RB1 deletion were correlating with the higher induction failure in multivariate analysis. So based on these copy number alteration profile, we can identify cases with good outcome as well as poor outcome. In the pediatric cohort also, again, the common deletions are in CDKN, most common, followed by PAX, Icaros, ETV6 and RB1. And we can differentiate patients with good outcome and poor outcome based on the copy number alteration profile around all the genes or only the Icaros. So if we use the Icaros deleted versus other cases, again, we can differentiate patients with poor outcome and good outcome. In large studies on pH positive BALL, again, we find that these alterations are common. In fact, more than 75% cases of pH positive BALL will show Icaros deletion. And these patients have poor outcome and resistance to chemotherapy, even in the patients treated with TKI. So in the pre-TKI era as well as in TKI era, these cases with Icaros deletion respond poorly. Now, currently in BALL, the management is largely guided by the flow cytometry based MRD assessment at the end of induction chemotherapy. But we found that many cases which are MRD negative, they are also doing poorly. So we analyzed in these MRD negative intermediate risk group, what is the copy number alteration profile? And we could see that the cases with good genetics versus poor genetics, we could identify patient even in the MRD negative group. So this can provide complementary information that in MRD negative group, if the baseline genetic profile is bad, the outcome is going to be poor. So don't just rely on the MRD or by flow. We can combine this information of copy number alteration profile to identify cases with poor outcome in this group also. So coming to the molecular genetic risk classification based on this copy number alteration profile, how can we integrate this information in the current management? So this was first proposed by Moorman et al. In two-tier genomic classification. What they did was they integrated this copy number alteration profile of these 8 genes, which are done on MLPA, with the cytogenetic information. So in the cytogenetic intermediate risk, they said that look at the copy number alteration profile. If the copy number alteration profile is good, they should be taken as good risk genetics. If the copy number alteration profile is intermediate or poor, then these cases should be clubbed along with the other high risk genetics. So overall, there should be 2 groups. One is good risk genetics and one is poor risk genetics. So this was the 2-tier genomic classification. And they validated this in their cohort to say that most of the intermediate risk group patients now can be classified into either good or poor risk genetics. So a lot of our patients also belong to this B other ALL category. They can be taken as good risk or poor risk based on the copy number alteration profile. So these were the criteria proposed by the Moorman group. For example, if there is no deletion of these 8 tested genes on MLPA, then it indicates a good copy number alteration profile. Whereas if there is any deletion of Icaros, pseudoautosomal region gene, early B cell factor or RB1, that will indicate a poor risk genetics. So this way, we can divide patients into good risk and poor risk genetics. Now in the German BFM study group, based on their 1000 BLL patient, they described another subcategory of patients, Icaros plus. This is defined as cases with deletion of Icaros, co-occurring with at least one additional deletion in CDKN2A, 2B, PAX5 or pseudoautosomal region gene, in absence of ERG deletion. So if we remember, the cases of DUX4 rearrangements or ERG deletions had a good outcome even in the presence of Icaros deletion. So these cases are left out of Icaros plus definition, and these cases have a very poor event-free survival as compared to cases without Icaros deletion. So this was the criteria were given by the German group, Stanila et al. In our cohort also, we found such cases where there is Icaros deletion along with CDKN and paired box gene. So they are labeled as Icaros plus in absence of ERG deletion. And these patients of Icaros plus subtype have a much worse outcome in the BLL cohort as compared to other cases. So now there are 2 types of criteria. One was Moorman's criteria, and one is these Stanila criteria for risk classification. So we wanted to combine the information obtained by these 2 types of risk scoring systems. So we added the 2 and proposed another molecular genetic risk scoring system, Molecular Risk Plus, which combines the information obtained by both these classification. So for our BLL cases, what we do is we give a score of 0 or 1 based on the Moorman's criteria. And we also give another score 0 or 1 based on the IKZF1 plus profile. And then we combine the score by both these classifications and get an overall score of either 0 or 1 or 2. So what we found that the outcome of these BLL patients, we can find patients with good, intermediate, and poor outcome, which is statistically different subgroups in terms of event-free survival, overall survival, as well as the relapse risk. So we see that the 4-year overall survival is 68% versus 48% versus 27% in these cases with MR plus score of 0, 1, or 2. So we can say there are 3 categories with low risk, intermediate, and high risk based on these MR plus score, which can be done at baseline using MLPA. Here we see the comparison of the separation of patients of good, intermediate, and poor risk based on the MR plus risk score 0, 1, 2, and the currently used ICICL risk protocol, that is the Indian Childhood Leukemia Risk Protocol. So the separation is not very good into standard, intermediate, and high risk. And this is mainly taking into account the MRD by flow at the end of induction, date, prednisolone response, bulky disease, and other features. So maybe in the next phase of the ICICL trial, this copy number alteration profile can be integrated for better risk stratification. Then we are also in the process of validating a weighted molecular risk score. That these copy number alteration information can be combined with the clinical information like the age and TLC, and then a weighted molecular risk score can be given. So what we saw that age more than 10, CDKN deletion and Icaros deletion get a higher score as compared to other features. And this risk score, weighted risk score, can give us the prediction of the first event over time. So if the risk score is high, the probability of first event over time will be much more. So these kind of nomograms can be prepared. So we have to validate this weighted molecular risk score in our prospective cases. Recently, we also combined our cases and did a collaborative study with the PGI Chandigarh on pediatric BLL patients on ICICL protocol under the guidance of Dr. Moorman himself. So we had a combined cohort of 493 pediatric BLL cases, which were on ICICL protocol. And as expected, almost 2/3 of our cases belong to B other category, where the classical aberrations, structural and numerical, could not be found. And almost 60% of our cases had at least one copy number alterations. And the proportion of copy number alteration tend to increase with age, and the prognosis tend to become poor with increasing number of copy number alterations. So here we see that the proportion of copy number alteration increases from standard risk to intermediate risk and to high risk ICICL categories. So the building tends to be longer. Similarly, in the cytogenetic good risk versus intermediate versus high risk, we see that the proportion of copy number alteration is much more in high risk genetic category. And the common alterations are Icaros, CDKN, and paired box gene. So to summarize, BLL is genetically heterogeneous disease. Latest genomics data has led to the identification of multiple new genetic subtypes of BLL, including some targetable lesions also. Copy number alteration based risk score, especially Icaros plus and MR plus molecular risk scoring, can help in better risk stratification and prognostication of BLL cases. And multiple diagnostic techniques may be required for detailed genetic assessment in BLL, maybe in a sequential way. So this is just to summarize that whenever there is a case of acute leukemia BLL, which is confirmed on flow cytometry with B lineage blasts showing CD19 and 79A, one can do cytogenetics for the ploidy and recurrent translocation. More commonly, recurrent translocations are now checked by either FISH or RT-PCR. Flow cytometry can be used for the ploidy analysis and for CRLF2 overexpression in these blasts. Then we can use FISH probes, break apart probes for multiple pH-like lesions. MLPA can help in detecting the cases with Icaros deletion, Icaros plus profile. Archer NGS can help to know the non and novel fusions in the 81 genes on the panel, which is commercially available. or we can do the transcriptome analysis for all the known and novel fusions. And for any novel fusions detected, we have to do orthogonal testing by either RT-PCR or Sanger sequencing based technique. So I thank my institution, all the funding agencies, and my students in the lab who have done all the lab work which has been shown in this presentation. Thank you very much for your patient listening.
Thank you so much, Dr. Sanjeev Gupta. That was fantastic. A lot of information, learned a lot.
Thank you so much.
So let's open up your presentation to discussion. And maybe you can use raise hand signs, all the discussants. So we can go in the order of as you put up the hand raise sign. Yeah, so we have, what is that, Roma LT? Okay, go ahead.
Thank you for so much elaboration of all this plethora of genetic abnormalities, sir. That was wonderful talk. Listening to all this, I'm little worried. So do we need to test for these genetic abnormalities initially for all BLL patients, or we can do it for those who are not responding well to the standard therapy? How do you advise?
Yeah, so I share your worry in the lab. So it is going to be a sequential assessment, I believe. So in the initial analysis, we may have 1/3 to 30 to 40% cases where we'll find the recurrent chromosomal abnormalities or ploidy changes, and they will fit in and respond to the standard chemotherapy. But the cases which are not responding, which belong to the B other ALL category, especially the clinical requests are coming for those cases which are not responding well to the standard chemotherapy, with MRD being positive, or they are relapsed cases. So there the clinical team is also wanting to know what are the lesions, whether they belong to PH like ALL. So if resources are available, I would say that everybody should be tested at baseline so that we know upfront and we don't lose any time. But to optimize resources, this is one of the strategies which is currently being followed. Dr. Agarwal can also share his thoughts on this, whether this should be a sequential thing or upfront at
Fair
baseline.
enough, you summarized it beautifully. Similar concern has been shown by many people in the chat box. Dr. Qadir Ahmad has said, do you look for all these abnormalities in daily routine? And then there is Dr. Kannan asking day to day, what is the minimum amount of tests that you will advise? Maybe these are the two questions which are very similar. You want to add anything, Dr. Sanjeev?
So the initial assessment in our setup also is that first the sample is tested with a multiplex PCR, which is checking for BCR-ABL, the translocation 119, MLL rearrangements, and 1221. And in addition, we also do a multiplex PCR for BCR-ABL. So that will detect P190 as well as P210, because in the multiplex, we may miss the P210 type of BCR-ABL. So these two panels of multiplexes will identify all your pH positive cases, translocation 1221, 119, and MLL 514 positive. In addition, we are also doing translocation 1719 by PCR currently, and we are organizing for a FISH probe. So 1719 has been included and has a poor risk subgroup. So that can be tested initially upfront. And when we do the FISH for translocation 1221, that will screen your cases for IAM21 also, because the same probe can show extra signals on RUNX1 using the ETV RUNX1 probe. So this is the initial panel. In addition, while doing the flow cytometry, we also test one tube for the FCV for ploidy analysis. So that complements, and CRLF2 is added in the same tube. So CRLF2 overexpression is done in that tube. So this can be the initial assessment. Later on, if required, all these break apart probes for pH like, they can be used, like ABL1, ABL2, MLPA. So they can be used depending on the resources and the response to the standard chemotherapy.
Maybe for some of the cases, CD10 negativity can help to
Yes. So
test
right now,
further.
correct. So right now, whatever comes CD10 negative, we put CD15 and NG2 to look for MLL kind of case. But now we can take up these cases, especially if CD13 and 33 is also present along with CD10 negativity. we can take up these cases for ZNF384 and MEF2D rearrangements and NUT-M1
Nothing.
and have these 3 break apart probes and test.
And is there a targeted therapy for them, sir, for these NAT-M1 and all?
Most of these are under trials, but there are some case reports basically where they have shown pH-like lesions being treated with the available TKIs. But large-scale clinical trials are still ongoing.
If I can continue, I have 2 more questions.
Go ahead. Go
So,
ahead. Go ahead.
yeah, the CRLF2 can be assessed through IHC as well, other than the flow cytometry. And is there any cutoff for percentage of blast to express for CRLF2 to take it as positive? Is
So
there
my
anything like
experience on CRLF2 is with flow cytometry, MLPA, and FISH. So I haven't done IHC for CRLF2, so I won't be able to comment on the IHC cutoff or the utility.
Okay, flow cytometry, is there any cutoff to call it as positive? These many blast expressing?
No, we look for a
Nothing like-
distinct clone. The numbers don't matter. So whatever cases show a distinct clone, it may be a small clone also with clear-cut overexpression beyond the negative internal lymphocytes. Then
Oh.
we can take these cases on qPCR or further testing. So
Okay.
there's no cutoff of percentages as such.
Okay. There is one point you have mentioned, gain of chromosome 21 with Icarus IKZF1 deletion, and there is a here amplification of 21. So between these two, how can we say this is because of IKZF1 and this is because of amplification 21 clear cut? Like both
Okay,
of
so
them
that-
have the gain.
Yes, so that combination was with IKZF1 and 159Y, where we said that almost 75% cases may have a extra chromosome 21. So that gain was an extra chromosome, like a trisomy. Whereas in intrachromosomal amplification, what is happening is there is a chromothripsis leading to alternate loss and gain pattern on the long arm of chromosome 21. So it is not the entire chromosome which is getting duplicated. It is a partial loss and partial gain in that chromosome.
Okay, thank you so much, sir.
Okay, welcome.
Thank you. Next in order, I do not know the name. It reads iPhone. So whoever you are, you want to ask your question? Yeah, go ahead. You're muted. You're muted.
Okay. Yeah, sir, am I audible now?
Yes.
Yes.
Yeah. Sir, wonderful, excellent presentation. And I will take time to assimilate what all has been taught to us by Dr. Sanjeev. Mind blowing. So, sir, I have a question. Like, what is the time for doing copy number variation? Is it post induction, even if MRD is negative, or further?
Okay, thank you for your compliment. And regarding the copy number alteration, the technique we are using currently is MLPA. Now the sensitivity of MLPA is limited. So we need to have at least 40 to 50% blasts in the sample. So that we will get at the diagnosis level at baseline. So we should test at baseline with a lot of blast in the sample, at least 40 to 50% blast. And this information, as we have shown, correlates well with the outcome. Whatever we do at the baseline, even if MRD is negative, they may have a poor outcome if the copy number alteration profile is bad. So baseline with a lot of blast, or when the patient relapses with at least 40 to 50% blast in the sample.
Okay, thank you, sir. I have another question. What is the percentage of various diploidy, hyperdiploidy or hypodiploidy in our Indian population? And what is the percentage of patients did very well when they had hyperdiploidy between 51 to 65? Because I read somewhere those patients have those patients shown some extra accumulation of methotrexate, which gave very good response to therapy and patient fared well. So do we have any experience in our this thing?
So there is a discrepancy between the Western literature and our Indian data regarding the prevalence of these ploidy changes. So there is a lot of challenge in, the cytogenetics, the culture, and the success of these cultures in our
Yes.
Indian setting. So we don't replicate whatever is the Western data, maybe
Yeah.
because of the techniques or, genetically also. But when we do flow cytometry, we find that the prevalence is quite similar when we do the ploidy analysis by flow. So maybe it has something to do with the cultures. We are not able to do a good quality culture to know these ploidy changes. Regarding your second part,
Yeah, that methotrexate accumulation within the blast is more when hyperdiploidy is between 51 to 65. That has been quoted in some Indian book authors. So I thought we can have direct from your mouth because you told so much detailed.
exactly, I won't be able to confirm that, but yeah, we generally take the hyperdiploidy cases as the good risk genetics. In fact, in the intrachromosomal amplification also, it has been seen that if high dose, high intensity chemotherapy is given, the outcome significantly improves. So maybe that is one of the mechanisms. I won't be able to confirm right now. So I will need to check.
Okay. Sir, one small question, maybe out of this thing, like in AML cases, when we see cup-like or fish mouth-like appearance morphologically, or if we see too much basophilia in pool of lymphoblasts, then we think, oh, this we have to go for BCR-ABL. Maybe it's de novo CML, or it is ALL with this thing, ALL with BCR-ABL positivity. So anything we have to look and catch in morphology, which will give us clue to narrow down our this molecular and cytogenetic workup, because monetary constraint is big thing to us,
Yeah, so as you correctly pointed out, like in cup-like blast in AML, we look for NPM mutations, and that helps. Like many times it correlates.
Yeah.
And looking at basophils, we get a hint that it might be CML blast crisis or, ALL with Philadelphia. That also works most
We
of the
got,
time.
yeah.
Yes. And other than that, flow cytometry generally gives us an idea, like
Yeah.
CD10 negativity to pick up those MLL rearrangement cases. Or if there is, a big blast morphologically, if we say that in cases with hyperdiploidy, the size of blast is slightly bigger. As
Very big,
compared
accumulate like that of AML, and at times we think could be that, and then when flow comes, it turns out to be BAL. Yeah.
Yes, with
So, thank you
hyperdiploidy. And
for your,
CD55 may also be almost absent in these cases with hyperdiploidy.
so
Similarly, if CD34 is absent, it correlates with translocation 119,
yeah,
right? So
yeah.
there are some flow cytometric correlation, like if we have BLL with CD13, 33, we check for BCR-ABL. So that is myeloid markers along with BALL. We also check for BCR-ABL in these cases. So generally we get on flow cytometry rather than morphology in ALL. And now we have more differentials like CD10 absent with 1333. We should look for
We,
ZMF, MEF2D,
you have
etc.
told, elaborate. Sir, any case you come across BALL, BCR, ABL positive, but P190 gene product? Did you come across
BCR-ABL
any?
with BLL with P190.
Yeah, yeah.
Yeah, in children, like the subtype will be mostly P190 type.
Yeah, yeah. We have got 2 110. Yeah, one.
So
Okay.
predominantly, yeah.
Okay. Thank you, doctor, so very much. And from you, take home messages, copy number variation, and again, look carefully our flow interpretation, especially CD10, DIM, and other myeloid markers to reach to appropriate cytogenetic testing. Thank you so very much. Yeah.
Ma'am, in WHO beta 2 version, it was given as 51 to 65 chromosomes, ma'am, to call it as hyperdiploid. So that is the correct.
Okay.
She wanted to know the mechanism, like why are they doing well? So I think that
Yeah.
is one of the mechanism, the accumulation of methotrexate. Yes.
Okay. Okay. Thank you.
From our side, we have tried to generate data on copy number alteration. So right now they are not the part of any management protocol, but maybe in the upcoming protocols, they should be adopted like with available data.
Thank you.
Thank you. We will be going to Dr. Jyoti Sarne, but before that, we'll take one question from the chat box. Is it possible to test close family members of cases of BALL for studies based on single nucleotide polymorphism?
I could not get the question exactly.
Is it possible to test close family members of cases of BALL for studies based on single nucleotide polymorphism?
So there are some germline mutations which have been described in Icaros and PAX gene. So those family members can be checked for these germline mutations. Like other than the N159Y, there are many Icaros mutations which have been found in the germline cases. So if these germline mutations are present in the family members, then it will become clinically relevant and they will become ineligible as stem cell donor in case the stem cell transplant is planned. So yes, there are some germline mutations in Icaros, PAX, and other genes which have been described, and they can be checked in the family members if they are present.
Thank you. Dr. Jyoti, your questions.
Hello.
Very nice lecture, Dr. Sanjeev. My talk is what all elements do you, my question is what all elements do you include in your report when you give this report to the patient? And how much does it cost to give this report to the patient? And if you're, do you give the score that you just spoke about? You have published a score. Do you include that in your report just to validate it? And just in case the score is high risk, does the clinician alter the treatment based on the high risk report that is he is given?
Yes. So what we are doing is that we are doing the known pH like fusions RT-PCR. So we give all the fusions which have been checked. Similarly, on MLPA report, we mention whether Icaros deletion is present and whether it belongs to Icaros plus profile. And then based on the other kit, the ERG deletions also, if Icaros deletions are there, in absence of ERG deletion, we mention whether this MR plus score is 0, 1, or 2. And it has been correlating clinically also. Like day before yesterday only, we were having a discussion like whether they are, deciding their management based on this. So yes, they were deciding their TKIs. So I got to know that whether they want to give dasatinib or nilotinib based on the Icaros positivity or negativity in a pH positive case of ELL. Although I don't know whether enough literature is there, but yes, they do look for these changes and decide the type of TKI they want to give. And they are correlating with the outcome also. Like in case where there is CDKN along with Icaros deletion, they prefer to give nilotinib rather than dasatinib. So this is what I got to know from the clinicians.
Thank you.
Thank you.
Thank you. Dr. Mike, your question?
So,
Hello.
thank you so much for your excellent talk, Professor. I am Yamala Shui from Myanmar. As I have two questions. One is the immunophenotyping for the diagnosis of ALL. As I am from the limited resources country, I would like to know which markers should be at least included for the diagnosis of ALL. And another question is, I'm concerned about the hematogones for the remission assessment from the bone marrow aspirate morphology. And is there any distinct morphologic features between hematogones and lymphoblasts for the remission assessments? As there is here, we cannot proceed the immunophenotyping for the remission assessment for that leukemia patients. Thank you, Professor.
So regarding the immunophenotype, so what we generally do is that there is a screening tube at the initial assessment. We look for the cytoplasmic marker, whether it belongs to the myeloid lineage based on the MPO positivity, or cytoplasmic CD3, or cytoplasmic CD79 along with CD19. So the initial tube guides us whether it is myeloid or T lymphoid or B lymphoid. So that way we can optimize, and if there is an ambiguity, we can do both these lineages, whoever are coming positive, and the initial cytoplasmic marker. So once we know that it is a B lineage leukemia with absence of myeloid and T lineage, then we look for the other B markers, which include CD19, CD10, along with other markers for MRD like CD123, CD58. So all these markers, CD73. So this panel includes markers which will indicate your initial diagnosis, which includes CD10, CD19, CD38, and which will differentiate later on with the hematogones and identify the MRD also. So these are the minimum markers for B lineage ALL, so that we can, as you said, the worry is to differentiate them from hematogones at the time of MRD analysis. So we should at least have CD45, CD10, CD19, CD20, and CD38 to see that, waterfall kind of pattern on flow cytometry. So that can help us differentiate from the hematogens. And specifically for MRD, we put additional markers, as I said, like CD123, CD58, CD73, and sometimes CD44 also. So this was the immunophenotypic panel.
Thank you
And
so much.
if CD10 is negative, then we have additional markers of CD15 and NG2 to look for MLL kind of rearrangements.
Sir, usually we keep for single tube for BALL with CD1979A and one myeloid marker like that. So by seeing these, I think we need to keep CD10 and 1333 also so that we can detect the.
Very true. So in that B tube, now we have to add CD13 and CD33 also. Because if there is a combination of CD10 dim or absent along with some myeloid markers, then we can go for ZNF384 and MEF2D. We may be missing a lot of these cases. Yes,
If we keep single tube, we will be missing.
yes, definite. Agree.
Okay, thank you. We'll be going to Dr. Geeta. And before that, I'll take one more question from the chat box. Dr. Hita wants to know, ALL with BCR-ABL P210 positivity, what is the percentage in your series?
So mostly in our adult cases, we have seen this transcript, like 50% will have P210 and 50% of patients will have P190 in adult subgroup. In pediatric, it is mostly like 80-20. I would say that predominantly it is P190 and around 20% cases have P210 type of transcript.
Thank you. Dr. Geeta, your question.
Yeah, thank you, sir, for the wonderful very explanatory type of discussion that you have given. I wanted to ask your routine algorithm for diagnosis, which you have just illustrated. Right now we do morphology, then flow cytometry, try to include CRLF. Decision on CD10 is now we have to change. Like before, we used to have only one option. One point that we could give the clinician to lead to further testing. So now we have to think of more differential diagnosis. And then, of course, FISH and further. So we rely heavily on cytogenetics and ploidy, conventional cytogenetics and ploidy. So just wanted to know, after listening to your comment on the discrepancy between the flow and the conventional one. So how, as such, it is a challenge. The conventional cytogenetics is a very manual, labor-intensive and challenging diagnostic modality. So now if over that, most of us, because want of this we rely heavily on this method. So what is the amount of discrepancy and plus what is the additional financial burden we have if we add the ploidy to the flow? These are my two queries, sir.
So I totally agree that cytogenetics is a labor-intensive thing, although it is cost-effective, but it has to be done well, and it is labor-intensive. It demands time and manpower. Regarding the flow ploidy by FCV, it is a dye, so it is just like DAPI. So it is not very costly, and just to gate the blast, you need a 45, 19, and 34, and 10. So these 3-4 markers along with FCV you can put in a tube. And CRLF2 can also be put in the same tube. So it is not very, very costly. It is cheaper than the other, fluorescent dyes, the other CD markers. This FCV is a non-fluorescent molecule. So it is cost effective, can be done as a supplement. And you will see that a lot of cases which are coming normal on conventional cytogenetics, you are finding ploidy changes on flow ploidy.
Right. So just one more point. Are you practicing germline mutation testing for all transplant donors?
Not right now, because the prevalence is very low. Around less than 1% cases may have germline mutation. So we thought of taking it up as a project, but right now we are not doing.
Thank you so much, sir.
Thank you.
Thank you. Dr. Deepak, you will be asking your question. And before that, one more question from chat box. Dr. Kanan wants to know, should we routinely ask for MLL break apart fish or multiplex PCR is sufficient?
So regarding the MLL, we are only testing for 514 on this RT-PCR. So we may be missing other rearrangements. If we can do fish MLL break apart, we are more likely to get more cases with MLL gene rearrangements. So for MLL, fish break apart probe is a better strategy as compared to the RT-PCR, definitely.
Dr. Deepak, your question.
You are muted, sir.
There's some problem, you just rectify that. We'll go to Dr. Umang in the interim period.
Thank you, Dr. Sanjeev, for such a great presentation and a lot of research. I'm very proud that it's been done in India, and we are almost near to US, what we always considered that they are in the first. But I'm really happy that we are doing so much of research and all these papers are coming from India. Two things. One thing already discussed, that MLL fish is always better strategy, and PCR we should know when we are ordering that either we are taking all the transcripts in process or not. If you are not taking, obviously ML fish is a great technology. Another thing is what I wanted to ask was, is there any fish probe available for your DUX4 rearrangement, which you mentioned that along with ERG deletions? Let's say we are not able to do MLPA right now, and if you want to identify such rearrangements, because the fish probes, we have the setup of cytogenetics and fish, but don't have the setup for MLPA. Is there any option for DUX4 rearrangement identification?
No, Dr. Umang, I haven't come across this fish probe for DUX4 rearrangement, so I really don't know if it is available in the market.
Okay. Thank you.
Oh, but for that, sir told IHC is available. Maybe we can get that IHC marker.
Fish probe, I don't know whether it is available.
Yeah.
Thank you. Dr. Deepak, are you ready? I'll take one question from the chat box. Does cesarean section, as compared to normal delivery, increase the chances of a successful second hit of ETV6-RUNX1 fusion triggered by infection in early infancy, as children born with cesarean are likely to have lower immunity as compared to those born by normal vaginal delivery?
I have no idea, sir. If you have any comment to make
No,
on
I have not come across this, so I can't answer this question. Okay, we go to Dr. Vibha.
Congratulations, sir. Excellent presentations and too many eye-openers. So of your entire presentation, what really got me worried and out of my comfort zone was the MRD paper of yours, which is showing that even your MRD negative patients, there is a dichotomy, the curve is separating over there. So two questions there. So, so do you feel, is it because we are still practicing flow MRD, or should we be graduating to better forms of MRD? So is the difference in the curves because of that? Or is it because we are missing genetic abnormalities? And in that case, what, again, it may be a repetition, but I really want to know that what we are already doing, what should we add, sir, keeping the cost in mind?
Oh, thanks for your question. Great question. So two comments to make regarding that curve, separating. One, maybe regarding that technique, flow is the best technique for MRD assessment. The specific translocations may not be there in many cases. So MRD is applicable to majority of the cases of BLL for MRD assessment, that is for sure. But are we doing it right? Are we doing it, sensitive enough assay? Because different labs may have different level of sensitivity also, different, assessment modules, or experience of the operator who is reporting. So depending on all these factors, it is variable. So we may be calling out cases which are actually MRD positive as MRD negative. That may be one component. But the major component is the disease biology. This is what I feel, that the patient may be responding initially, leading to MRD negativity, but ultimately they are going to come back if the genetics is poor. So that is going to be a minor component of the technology, like MRD, whether it is sensitive enough. But I think the major component is still going to be the disease biology, which we are missing, because MRD is just a surrogate of the total, genetics as well as response to therapy. So maybe these poor genetic subtypes have an initial response which is lost very quickly. So that
Then in that case, what should we add to our armamentarium?
What we have,
I mean, we are doing the fish and we are doing the flow ploidy and fish, and then we stop there.
So what we really wish is that we have generated enough data that at baseline, you should do this copy number alteration profile. If it is poor, it is poor. And you upfront decide whether to take a patient to the transplant. Just after achieving the remission, just as we do in AMLs, these patients have to be taken up for transplant or any other, clinical trial, maybe some experimental therapy. So once we know at baseline it is poor genetics, there is no escape, even if flow MRD is negative in the initial run or after 1 month. So that is my take on this.
Is there any data on that? I mean, taking up kids for upfront transplant based on the copy number? I mean, what is the
data
real
is like, if we are just following up on ICICL protocol, just intensifying if based on MRD, not intensifying if MRD is negative, they are doing poorly, as we have shown. The graph are splitting. So something needs to be done for these patients who are MRD negative, but ultimately they did poorly, and that was suggested at baseline by the poorest copy number alteration profiles.
So outside our country, from the world, is there, so are you aware of any data where based on these, which genetic profile they are taking up upfront for, after, of course, MRD negativity, taking up them for transplant upfront? So and which variation or which genetics? Sir, I'm a clinician, sir. Just a disclaimer, I'm not pathology. I'm a clinical pediatric hematologist.
Yeah, so I guess you need a lot of data, clinical validation data before you can
absolutely
use this information. So that is what we are trying. As a first step, we are trying to generate data based on the genetics and the outcomes. So now it has to be taken up in the trial setting, the RCT setting, where you can show that when taken up, these cases have done well. So this kind of validation needs to be done.
Yes. So presently, all the validation is based on the MRD, and probably they are doing NGS MRDs and all that. So that's what the data says. So
our findings and efforts are in that direction, that please just don't rely on MRD. A lot of MRD has been done. Now go one step ahead.
Thank you, Dr. Viva.
Yes, sir.
Dr. Deepak, are you ready?
Sir, can you hear me, sir?
I can hear you.
Yeah, audible.
Yeah, sir. Good afternoon, sir. It was an excellent speech and very crystal clear. And sir, my doubt is there are a lot of copy number alterations which are there now, numerous copy number alterations responsible for upfront, baseline diagnosis and also in MRD, relapse cases. So sometimes, even cases which are called, termed as good prognosis, like hyperdiploidy, if they have some mutations like CREB BP mutations with affecting HAT domain, they may not respond to corticosteroids. So there are few more examples. So taking all this into consideration, is it practical to make Archer fusion plex targeted RNA sequencing a mandatory test in all baseline cases of BALL among Indian population for better management of patients? And second question is, will this whole transcriptome sequencing replace cytogenetics and other molecular tests in near future?
So excellent questions again. So I would say that make it mandatory, but who will pay? The only thing will be the money, resources. So if we can make it available, if resources are there, definitely it should be done for all cases. Like the COG is doing for all their BLL cases, they are screening for all these lesions by this sequential approach, the low density array followed by Archer if required. So if resources are available, definitely. If not, then depending on whether the patient is responding, not responding, and sequentially whether we can do it. Regarding your other part,
Whole transcriptome sequencing, will it replace in near future?
I think all these techniques are complementary. If we can manage to hold on to the conventional cytogenetics also in addition, that gives a lot of information which may not be there on transcriptome alone, right? So we can actually see the chromosome. So the labs which have this facility, they have to hold on to this. So they are all complementary, I would say. Nothing is going to replace the conventional cytogenetics as of now.
Thanks a lot, sir.
RSEDH will replace, but that has not done so.
So at least hope they make this RNA sequencing panel, one of the tests, because many places they just do flow cytometry and conventional karyotype, and few tests like translocation 1221, 119, and MLL rearrangement, but they land up with relapse.
Yes. So what we have seen is that the cost is also coming down. So it will be commercially available now. The whole transcriptome sequencing is now affordable. And with more and more, ordering, the cost may further come down and may become affordable for the patient.
Thanks a lot.
Thank you. We'll take a question from chat box again. Do you advocate IGH IL-3 fusion studies in all cases of BALL having hypereosinophilia and lack of peripheral blasts?
So again, that is a sequential analysis. Once we have, done all the classic rearrangements, cases with eosinophilia, includes PDGFRA, PDGFRB, and all those translocations. So if that panel has been exhausted and everything is coming out to be negative, then only this is done. So this is not a very commonly done test in cases with eosinophilia, but yes, if required, can be done.
Right. Dr. Rupam, your question.
Rahul sir, thank you. As always, learned a lot from your talk, sir. So I have two queries. One is that does NG2 expression always correlate with MLL expression? Because we had one case few months back here at Guwahati, where based on the flow and the skin lesions and all, we suspected this may be BPDCM. So we sent one sample to extract where NG2 was positive. And so acute leukemia of ambiguous lineage was suspected, but MLL rearrangement was negative in that case, sir. So I wanted to know whether it always correlates with MLL or sometimes in your experience, it may not. And secondly, sir, some of these new genetic categories of BALL like DUX4 rearrangement, what I have read is that after induction and after therapy, they may show some monocytic markers, monocytic differentiation. So do you recommend that in these scenarios, better more than flow molecular testing is better for MRD assessment?
So regarding your first case that when we have CD10 negativity, we do NG2, CD15, everything falls and suggests that there could be MLL gene rearrangement. But when we do RT-PCR, we may not find it. So that is our experience also, that 100% may not be correlating, but then we may be missing. So as we said, that whether we have done FISH break apart probe or just a PCR for 2-3 fusions, that also can, make us show the correlation or miss the correlation also.
Yes, that is a break apart, sir.
You did the FISH also, and that was also that also we have seen many times that even with FISH, it may not be correlating all the times. And regarding the second part was, what was your second question?
Yes, sir. Some of these new entities like DUX4 rearrangement, after therapy, those blasts may show some monocytic marker expression. So do you recommend that molecular testing be more preferable than flow cytometric based assessment for MRD, because those monocytic markers may create some confusion in them?
For MRD, right, as we saw that in DUX4 cases, CD371 may also help. We are trying to include that also in the flow panel. So if we have a particular, translocation and we have got the primers, definitely it is more sensitive and more specific to do MRD analysis by those genetic markers. But if they are not available, then MRD is still the good backup, because based on those, MRD markers, which are differentially expressed in blasts as compared to the normal hematogones. So flow cytometry is applicable in most of these cases, especially with CD371. DUX4 cases can also be analyzed on flow. But yes, if we have got the primers for specific translocation, we can use that also.
Thank you, sir.
Thank you.
One question again from chat box. Dr. Sutanthira wants to know, do you use IGH gene rearrangements at diagnosis for all BALL for MRD monitoring, or based on initial diagnostic marker? Most of them detected by FISH may not be as sensitive.
So currently we are not using the IGH gene rearrangements or any other molecular gene rearrangements for MRD analysis, right? So we are mainly doing by flow cytometry only. The MRD is done by flow, even if we have these markers being present.
Thank you. Dr. Akshay, you had raise hand sign, you removed it. You have a question?
So my question was actually in continuation to the previous question only, that you have mentioned about CD371 as a surrogate marker for DUX4. So are you recommending it that we should incorporate it in our panel, because it's not a very common marker?
So I have ordered it for my project. So I would say you should also generate some data, and then we can talk maybe in the next lecture or conference, whether it works or not.
Sir, another question I was about to ask was about, have you encountered any case of switching of lineages in BALL? Like new WHO has mentioned about DUX4, ZNF384 and PAX5 mutations, where monocytic differentiation has been reported. Sir, have you encountered any such case?
We had one case of ALL, which after chemotherapy switched to AML M6 subtype, not the monocytic. So it is not very common in our experience, at least. So I just had one case in last 10 years, which, switched to AML M6. But yes, there may be many more cases outside of our reporting, but no personal experience of cases getting to monocytic and the ZNF384.
And sir, what is your experience with CD2? So because along with CD371, CD2 we are routinely using. So
it is not in our panel of BALLs. So generally we can't comment whether how many of our BALLs had CD2. So that is in the TALL panel.
Right.
Thank you.
Yes, sir.
Thank you.
Dr. Geeta, you have put a question, but you are there, so you can ask directly your question.
No, sir. I was just thinking that for the MRD negative category, which goes in for later, that it actually goes in for a poor prognostic issues. So then probably we should look at targeted therapies if we want to give the benefit to all our at least children. So that was just a thought, sir, that are we to move towards that?
Yes, definitely. If MRD is negative and the copy number alteration profile is indicating a poor outcome, then definitely these patients can be taken for RNA sequencing to look for specific fusions. And if some of them are targetable, then the patient will definitely be, getting those targeted therapies also.
Dr. Mahesh, you have a question?
Yes, sir. Thank you, sir, for giving me this opportunity. And I thank you, sir, for giving a very wonderful lecture as well as a very elaborative lecture. I had two questions. What is the role of NGS and at what point of time is it indicated in the workup of these cases? Secondly, should the molecular testing be repeated at relapse?
Okay, so NGS, as I showed the sequential strategies, that first we do the conventional recurrent translocations and ploidy analysis and CRLF2 everything. If everything is coming negative and the MLPA is indicating intermediate or high risk profile, then we take these cases for Archer-based panel. So this is our strategy, to look for the intermediate and high risk cases first, because to optimize resources, we can't do everything on NGS. So we have first taken up the 24 cases with high risk BLL, and out of that 5 showed these fusions. So almost like 18-20% cases of high risk BLL. So we wanted to see cases which are high yield for these fusions. some of which may be targetable. So ideally, all cases of B other ALL, especially if they are MRD positive or not responding, relapsed, all these cases should be done for NGS. But we are currently doing only the high risk based on the MLPA profile. So we do MLPA for all the cases of BLL. Wherever the MLPA profile is poor or intermediate, and they are not responding well, MRD is positive, then we take up these cases for NGS. And now we have started sending for transcriptome sequencing. So the results will be available after a few months, right? And the second point, I forgot.
Should the molecular testing be repeated at relapse?
Okay, at relapse, we have repeated MLPA in some cases, but the findings were similar. If Icaros deletion was present at baseline, it was present at relapse also. So there was not much of a change as far as MLPA was concerned or the recurrent translocation. If it was negative for all recurrent translocations at baseline, at relapse also, they were showing these negativity for recurrent translocation. So we haven't seen the changes at the recurrent translocation level or MLPA level, but definitely if we look at the genetic level, there will be additional mutations for which we have to do the NGS, DNA NGS or the RNA based NGS analysis. So there we will have some subclones which have increased in percentage, but that is not detectable by MLPA or conventional RT-PCRs.
Thank you.
So that we need to do single cell sequencing or the transcriptome analysis of the relapsed case.
Thank you, sir.
Thank you.
We're finished with one round. There are some raise hand sign. Any one of you have questions, you can go ahead.
Sir.
Go ahead.
Yeah. So flow cytometry, you have mentioned about the DNA ploidy index, right, sir? Is there any cutoff for hyperdiploid, haploid, and to call it as?
So all these are standard protocols. The numbers are there, based on which we'll say low hyperdiploid, high hyperdiploid, hypodiploid, normal. So all these cutoffs are already there, standardized, published.
Okay. Yes. And WTS, you have said it will take months. So is it we need to wait for months to get the report of that, or how many days? By how many
Generally,
days?
they commit 6 to 8 weeks for the sample to be, processed and analyzed. If you want to get the analysis also, if you have the bioinformatic department, bioinformatic pipeline within your department, the timelines may be lesser. But if you want to send and then get, then 6 to 8 weeks is the general commitment.
But in aggressive cases, waiting for that long, will it help really?
So you should send it as fast as possible, maybe at baseline if resources are there. So definitely we lose time if we do it sequentially.
Yeah, sequentially will not work in such cases.
Yes.
Okay. Thank you. Thank you so much, sir.
Thank you, Vibha.
That was so patient explanation of all. That is wonderful.
Yes, Vibha.
Yes, I want to ask, sir, is the MLPA for copy number variations and the whole transcriptome analysis commercially available now, sir?
Yes. MLPA kits are available. In fact, earlier there used to be a discount also for the institutions, but now like the DSS is marketing with the help of that. MRC Holland, that is the only company which is making these MLPA kits, and they are available in Indian rupees and commercially available, yes. For
and
ALL.
even the whole transcriptome.
Yes, whole transcriptome also can be ordered. A lot of companies take these orders nowadays. So you can outsource and get it
So
done.
is that same as the RNA?
RNA sequencing, yes.
RNA sequencing, is that same, sir?
Yes, so the Archer panel is also commercially available, but the limitation is it has only 81 genes. So we can get to know only the fusions of these 81 genes. But if we want to know all the genes to be covered, then RNA sequencing can be sent. I think 20 to 30,000 is the average cost these days, for which you can get the RNA sequence through the fusions.
And so what is the difference between DNA sequencing and RNA sequencing? Where do we order which one?
Right. So you must have seen in AMLs, it is very, very common to get NGS done. So that is all based on the DNA, because the point mutations are very common and clinically relevant, like IDH1, IDH2, FLT3, duplications. All these mutations are point mutations which are tested on DNA. But RNA is more labile, more sensitive, difficult to do. in ALL, we need RNA sequencing because there are fusions. PH-like cases also, because recurrent fusion, we need to check the transcript, the RNA transcript. That is why we need to do the RNA sequencing if we have any recurrent fusions in mind. Like BLL, the most relevant are the fusions, recurrent fusions. So RNA sequencing is more helpful. DNA sequencing role is very limited in BLL.
Thank you.
Thank you, sir.
Anyone else has a question? Okay. So in that case, Dr. Sanjeev Gupta, thank you very much.
Thank you.
Wonderful lecture and a great discussion. It was a tremendous
sir.
learning for me, and I'm sure it must be very, very good for everybody who was there today. So
Thank you.
thank you for sparing your time on a Sunday, and bye-bye to everybody.
Bye-bye. Thank you.
Thank you, sir.
We have
And
Intas here for official vote of thanks. Just hold on. Intas, are you there?
Great questions, all of them. So I also learned a lot. I have some homework to do.
Okay, I think Intas has logged out. So, well, that's the end of it. Thank you very much. Bye-bye.
Thank you so much, everyone. Thank you.
Thank you.